So our second speaker is Katie. Sorry, I'm probably gonna pronounce your name wrong. Katie Ayasoufi. Is that
That's
right?
correct.
Okay, thanks. So she's a research associate at Mayo Clinic in the lab of Dr. Aaron Johnson. She has been working on several projects related to brain immune system crosstalk during neurological diseases for the past 5 years. And she's about to become a PI, which is very exciting. I assume she'll be hiring soon. So if any of you are on the job market, please reach out to her. And yeah, so let's welcome Katie, and please take it away.
Thank you so much for the kind introduction. I'm really happy that I got this opportunity. So I thank the Open Box Science organizers for giving me the opportunity to present our newest preprint data relating brain resident memory T cells and their responses during CNS injury. Resident memory T cells are a subtype of memory T cells. However, unlike effector memory T cells that can circulate within non-lymphoid tissues, and blood, and central memory T cells that can only circulate between lymphatics and blood, these cells circulate solely within the non-lymphatic tissues. They persist for long periods of time, and they are actually estimated to be the largest memory T cell population in the body by number, both in humans and mice. They provide very rapid and organ-specific immunity. This is very important, especially in barrier tissues, including skin and mucosal membranes, where you need a very rapid response against pathogens. But the role of TRMs and other immune organs, including brain, has been very coming forward, and it's been emerging. However, there are gaps in the knowledge when we're specifically talking about brain TRMs. And these are the goals that we are trying to address in the study. So the first one is, do the healthy mice in the brain at homeostasis have TRMs in their brains? Can these brain TRMs actually respond to neurological insults? Can a TRM that resides within the nervous tissue detect that there's a disturbance and actually respond? And finally, does the direct involvement of brain TRMs with our antigen or during brain injury can actually induce neuropathology within the brain, or actually alter immune system responses outside of the brain? So these are what we will be focusing our studies. To begin with, we asked a very simple question: does the naive mouse brain contain resident memory T cells at baseline? To do this, we perform intracardiac perfusion in the naive mouse. So this mouse has no brain injury. And we use a large volume of PBS to actually clear the vasculature through perfusion, so that the brain, when we isolate and homogenize and use for flow cytometry, can only contain T cells within the parenchyma. We did high parameter flow cytometry, and as you can see here in the gating strategy, once we gate on all cells, single cells, live cells, we have two populations of immune cells within the nervous system that are distinguishable by expression levels of CD45. You can see that this high expressing CD45s, they are the traditional immune system that can come from the outside into the nervous system, while these intermediate expressing CD45 positive immune cells are actually CNS resident, in this case, likely microglia. But once we gate on these infiltrating immune cells, we can clearly see a population of T cells. They're actually both CD4 and CD8, both populations can be found. And within those, when we focus on the gold standard marker of resident memory, CD69, we can actually find that they have high expression levels of CD69. And this is important to show that within the brain, actually, majority of the T cells that are found in the naive brain express high levels of CD69, being consistent with resident memory, as opposed to the spleen, which is not supposed to have a lot of resident memory T cells, and accordingly has very few T cells that express CD69. So we then went further, and we really wanted to prove that these T cells that we find in the naive brain are indeed within the parenchyma of the brain and not within the vasculature. So before we did our perfusion this time, we actually used the gold standard method that immunologists use to say a T cell is within the tissue or within the vasculature, using IV labeling. We used anti-Cy1.2 antibodies, which is broadly going to mark all of our T cells 3 minutes before euthanasia. So we had the mouse that we injected intravenously with this fluorescent antibody. We then proceeded with our perfusion and isolation of the brain. with the idea that if a T cell has the fluorescent marker, it could have only gotten it through this intravenous injection, and hence this T cell was actually within the brain vasculature or close to it. Whereas if our T cells that we find in the brain do not have this marker, then they are confidently actually within the parenchyma and not near the vasculature. So once we perform this, and you can see compared to our negative control, which is a mouse that didn't get an injection, majority of these T cells that we find in the naive brain are in fact within tissue parenchyma, as they do not stain with the IV label. In contrast, in the spleen, where it's expected to have a lot of T cells still within the vasculature even after perfusion, we can confirm that spleen indeed has lots of T cells that have IV positivity, but brain does not. And then we further characterize these T cells that are found in the naive brain. And as shown before, they can be either CD4 or CD8. They do not express this IV label. They express high levels of CD44 and low levels of 62L, being consistent with a previously activated cell that doesn't go back to the lymphatics. They express high levels of CD69, being consistent with resident memory, and varying levels of CD103. These two markers are traditional markers of resident memory T cells, concluding that yes, the naive brain contains resident memory T cells. Moving forward, we next ask, then, can these T cells that are found in the naive brain that are existing there before we even deliver any insult respond to an acute insult? For this, we use a neurotropic virus infection, in this case, TMEV infection. Tyler's murine encephalomyelitis virus is a neurotropic virus that directly infects neurons and can propagate on its own within the brain and cause massive infection. However, in this case, we picked 24 hours as our time point to evaluate responses of existing TRMs. We did this because 24 hours is too early for priming and generation of a typical immune response, so that at this point, it's likely that what we see in the brain is isolated responses for existing TRMs that were there before the injury and not new T cells that are coming in. So when we did this, we first focused on the CD69 positive, CD103 negative population, because this was the major population we found in the naive brain. So as you can see here, 1 day post-infection, this viral infection can actually induce significant expansion of both CD8 and CD4 TRMs within the brain. For the sake of time, I'm not showing you the rest of the data, but this isn't virus specific. We have repeated this with physical injuries to the brain as well as a cancer insult. And in all cases, we can see this early expansion of TRMs that occur within 24 hours of the insult. So we then ask, okay, do these brain TRMs actually, do we have different waves of CD69 positive and negative populations, or are all TRMs suddenly expanding within 24 hours? So to actually answer this question, we decided to not only show our 24-hour data, but also extend beyond that and look at different time points post-infection to show what is happening to TRM populations within the brain parenchyma. When we did this, we can actually found that this early expansion of TRMs that occurs at day 1, which is very drastic, is mostly and exclusively to these CD69 positive, 103 negative TRMs, which is the majority of resident cells in the naive brain. So they expand rapidly by day 1. However, the responses and expansion of CD69 positive, 103 positive TRMs actually occurs at typical time points where we expect an adaptive immune response in the brain after day 8. These, however, persist even after virus is cleared, which is around day 30 days, you can still find them. We will think that these are likely antigen-specific cells that were generated after the infection. So that brought us to the next question. Is this early expansion of brain TRMs within the brain that occurs 24 hours antigen-specific? So to answer this, we used our viral model. Because we know in this model, majority of the generated CD8 T cells are actually against one epitope. This immunodominant epitope, VP2, actually is over 90% of our response in this viral infection. And we have tetramers for them that you can actually stain T cells for, and at various time points evaluate how many antigen-specific T cells are in the brain. So when we did this, when we focusing on CD103 negative or positive cells, these early time points when we saw this TRM response and expansion, we actually do not have any antigen-specific cells at these time points, either 103 positive or negative. They actually start coming into the brain beginning on day 5 and later on. Interestingly, the CD103 positive TRMs come in later. Their expansion occurs after the 103 negatives, and they actually persist long after the virus is cleared, indicating that actually there are two different waves of TRM responses within the brain, one that's 103 negative and one that's 103 positive. So we then ask, is this expansion of TRMs that occurs very rapidly and appears to be antigen non-specific result of proliferation within the brain parenchyma, or perhaps infiltration of some new T cells because of the injury that come into the periphery? To answer this question, we use this drug FTY-720. FTY-720 blocks T cell migration out of the lymph nodes and spleen, and it has been established to completely abrogate responses between the lymphatics and the T cells. So while the T cells do not die, they cannot move from the secondary lymphoid organs, and therefore in this model, they cannot go to the brain. So in this case, we then started the FTY before infection. We then started our intracranial infection on day 0. And at the same time, simultaneously, we delivered BRDU injection. So this is a dye that, this is a molecule that will intercalate into the DNA of proliferating cells. So if a cell is proliferating, we expect it to be BRDU positive. And at the same time, just like before, we administered our intravenous antibody that labels T cells in the last 24 hours, so that when we look at the data 24 hours later, if the cells have this IV antibody, that means they're infiltrating, whereas if they have the BRDU and not the IV, they're actually proliferating within the brain. With the bottom line being, if the FTY can block the early TRM response, then the expansion is due to infiltration. But if it doesn't change outcomes, then the early expansion can only be due to proliferation within the brain. So when we did that, we first performed high parameter flow cytometry with many different immune markers and tried to globally look at differences. And as you can see between the naive, where it has very few immune cells here, both the infected mice have significant amount of immune infiltration. However, when you focus on the control infected versus FTY treated, there's no global differences that we can see at this level. We went further and analyzed BRDU positive T cells. So again, these are the T cells that are proliferating within 24 hours. The naive mice have very few T cells in the brain that are proliferating, whereas both infected and infected plus FTY treated groups have significant numbers of proliferating T cells. This was quantified. So again, when we quantify expanding T cells, FTY really did not block this expansion. And you can truly say that BRDU positive, C9 positive, CD4 and CD8 T cells are expanding within the brain with and without FTY. And conversely, when you look at infiltrating T cells by our IV label, their frequencies did not increase, indicating that this TRM proliferation early in post-viral infection in the brain is due to proliferation, as FTY did not change outcomes, and that there's really no evidence of enhanced infiltration this early into the model. So with that, I summarize the first part, that the naive brain harbors populations of TRMs by phenotype. They actually expand as a result of CNS injury or infection very early, within 24 hours. I've shown you two waves of TRMs, one that occurs early, which is the 103 negative population, which is then followed by a CD103 positive TRM response within the brain. I've shown you that the expansion of brain TRMs really precedes infiltration of any antigen-specific T cells, and that this expansion we see 24 hours post a CNS viral infection is really due to in situ proliferation within the brain and not infiltration of immune cells into the brain. However, I did show you that despite this early response, we do get a really good antigen-specific response that really persists after viral infection. So in the second part of the talk, we really want to focus on that response by asking two broad questions. Can TRMs that are persisting in the brain after viral clearance be reactivated? And if they can be reactivated in an antigen-specific way, What is the consequence? Do they cause neuropathology in the brain? And can they change systemic immune responses outside of the brain? To do this, we developed this novel method of reactivating TRMs in an antigen-specific way in our laboratory. So we first infect the mice and allow 8 weeks for clearance and memory generation. We do know from many publications in this field that this black 6 mouse completely clears TMEV within 21 to 28 days later. So by 8 weeks, we're a month after viral clearance. So we're really truly looking at resident memory responses. We then give one peptide, either irrelevant peptide E7, which has nothing to do with this virus, so should not reactivate TRMs. Or we give the immunodominant peptide VP2, which actually does reactivate TMEV-specific TRMs. We then do, as usual, our IV injection in the last 24 hours and look at the responses later. So I want to bring your attention to the advantages of this model. We really think this model has extreme advantages over whole pathogen rechallenge because the peptide reactivation will really isolate CD8 responses away from other aspects of memory, even those of CD4 T cell responses. Additionally, neuropathology that will be observed in this model can be directly and causally linked to responses of CD8 TRMs alone. And that we know that neutralizing antibodies in this model cannot interfere with TRMs responses because there's no antibody against such a small class 1 fitting peptide. And this has been verified by us and others in the field. And finally, reviral infections or pathogen rechallenges with viruses can cause their own damage directly to the brain independently of TRM responses. And this model really gets away from that. And we can say that if there is damage, it was really caused by the TRM alone. So with having that in mind, we then specifically asked, does antigen-specific reactivation of TRMs causes neuropathology within the brain? If it does, what it looks like, and also can antigen-specific TRM reactivation then be considered a neurological insult that can induce peripheral lymphopenia? This idea comes from our previous work by us and others that have shown any brain injury, whether it's acute or chronic, can have consequences, including lymphopenia outside of the brain. So this is what we wanted to test. So again, we gave our reactivating peptide, and then we evaluated the responses. We focus on the brain pathology on 3 specific outcomes. One is overall infiltration of immune cells into the brain. You can see compared to a naive brain or a previously infected quiescent brain, reactivated brain with TRM reactivation can actually increase this infiltrating immune cells. Additionally, focusing on inflammatory monocytes, which are a huge population in neuropathology, we can actually see also a significant increase when TRMs are reactivated compared to the controls. And finally, we focus on CD8 T cells in the brain themselves. If they are larger by side scatter, it has been attributed that these CD8s are pathogenic and actually very activated. And again, you can see compared to both controls, these TRM reactivation increases activation of CD8s in the brain. So this is what's happening in the brain. This evidence provides there's neuroinflammation within the brain. And when we look in the blood, conversely, TRM reactivation induces significant lymphopenia, as you can see here, indicating that antigen-specific reactivation of memory T cells is a true brain insult that induces inflammation in the brain and causes severe lymphopenia outside of the brain. So we then ask, does this neuropathology that's associated with the markers that I just shown you also manifest with blood-brain barrier disruption? So as you can see here, we did a T1 weighted MRI where we injected gadolinium, which is a large enough dye that on its own cannot cross blood-brain barrier. But if the blood-brain barrier is leaky, it can go in the brain and be detected. You can see here the controls that had previous viral infection but are not being reactivated do not have leakage, whereas the VP2 injected animals, so when the TRMs are reactivated, this neuropathology is indeed associated with detectable and significant blood-brain barrier leakage. And interestingly, this extent of this blood-brain barrier permeability correlated with the extent of lymphopenia, again, linking these two phenomena that once you do something to the brain that causes pathology, you also affect T cell responses outside of the brain. And finally, because we know that this TRM reactivation with peptide is systemic, we really wanted to narrow down which T cells are mediating the response. Is it the brain TRMs or perhaps some other resident T cell in the lymphatics that can come to the brain and cause pathology. To address this question, we use two parallel strategies. One, we developed this dosing of anti-CD8 depleting antibody that gets rid of antigens or all CD8s, but outside of the brain. If you give it at an enough dose, it doesn't cross the blood-brain barrier and it cannot deplete TRMs in the brain. We then do the same model with TRM reactivation and ask what happens to pathology. In parallel, as before, we used FTY720, which doesn't deplete T cells, but it causes them to get sequestered within the lymphatics so that they cannot infiltrate the brain. So they both address the same question. So as control, we confirm that indeed these treatments are doing what they're supposed to. Within the blood, we do not have any circulating CD8 T cells left, either with depleting therapies or FTY. In the spleen, the CD8 T cells are indeed depleted, whereas FTY, as expected, does not deplete them, but does sequester them. And importantly, within the brain, none of our treatments are affecting numbers of CD8 T cells. In fact, they're exactly the same as our control, indicating that TRMs in the brain are left intact. So, and then in this model, when we reactivated them, we asked what happens to the same facets of neuropathology. What we found is that depletion of CD8s from periphery does not change the overall neuroinflammation by infiltrating immune cells, numbers of monocytes, inflammatory monocytes, or activation of CD8s, indicating that these responses are in fact mediated by brain TRMs and not T cells outside of the brain. And FTY treatment produced similar results, where when you sequester T cells, you still get same amount of neuroinflammation upon TRM reactivation by all three of these marked things with inflammatory monocytes, CD8 activation, and total infiltration. And finally, we asked, what about the MRI? Can we still have the leakage and the blood-brain barrier permeability even when we don't have any CD8s outside to respond? And indeed we do. As you can see, there is blood-brain barrier leakage here, whether you're looking at just TRM reactivation or TRM reactivation within the brain, we can see similar neuropathology. So to summarize, I've shown you that the reactivation of brain resident memory T cells leads to neuroinflammation within the brain, which can be part of any of these, including inflammation associated with inflammatory monocytes, activation of T cells within the brain, and blood-brain barrier permeability that's actually detectable by MRI. I've also shown you that that's not just what happens in the brain. You can actually have antigen-specific TRM reactivation that leads to peripheral lymphopenia and changes outcomes in the periphery. And it actually correlates with the severity of the blood-brain barrier permeability. And that finally, this neuropathology post-antigen-specific TRM reactivation is really the result of TRM reactivation within the brain and independent of actions of other resident memory cells outside of the brain. And I think we are really the first ones to show this point definitively. So what I've shown you overall is that the healthy brain has some TRMs during acute insult. These TRMs respond very rapidly within 24 hours, can detect the insult and expand. However, they then recover and generate only an antigen-specific population in the brain that upon antigen-specific reactivation can cause as neuropathology associated with immune infiltration, blood-brain barrier disruption within the brain, but outside of the brain, it also causes lymphopenia in the blood compartment. With that, I want to thank everyone in my lab, my mentor, our collaborators, as well as all of our funding sources, and I will take any questions. Thank you so much for your attention.
Great, that was an excellent talk. Thank you so much, Kathy. So we have some questions in the chat, so I will start with those. The first question is from Yushan Wan, and they ask, will the T cell in the blood enter the brain after FTY720 treatment and also the meninges?
That's a great question. So we have verified by our IV label that there's not an increase in infiltration with FTY. So we don't think that they're coming from the blood. And we actually showed that in the blood, we have close to 0 numbers of CD8 T cells left after FTY treatment. So certainly they're not coming from the blood or in the spleen. We have verified that. Meninges is a hot topic. No one actually knows if infiltration of T cells from the meninges into the brain is or is not affected by FTY. We don't think it is because we would expect them to take on the IV label, especially in 24 hours. However, that's pure speculation, and I have really no experimental way of testing that. So I can't exclude that meninges isn't contributing, but we're very sure that the blood and lymphatics are not part of this, and it's really the resident TRM.
Okay, great. The next question is from Xinjiang, and it's a question I've also been asking myself. It's what is the function of TRMs in CNS? And also because you see this very rapid response, right? Are they like the sensing or the first line of defense or something? Yeah. What is it?
Very great question. Also a reviewer question. So what we actually think the function of the early, so what I've shown you is two different TRMs. One is the early one that rapidly expands. And we think that cell type is important for bringing in innate immune cells and other immune cells. We have some strategies in our mouse where in our animals, in our lab, where we can actually get rid of MHC class 1 conditionally. And we can show that if these TRMs are gone through those strategies or they don't expand, we have less infiltration of innate immune cells, even if your rest of the adaptive immune cells in the periphery is intact. So we think the function of those early ones is actually to bring in cells that can clear the virus. And we think that's the hallmark of them is that they're antigen non-specific. So that's why they respond no matter what injury we give, because they detect something is wrong and they need to bring in the immune system. Now, the second part, which after infection, they get generated and they're actually antigen specific. So that would be a typical immune response we expect them, so that upon re-challenge, they would clear the pathogen, which is good in any other immune organ. But we would argue that in the brain, any damage is irreversible. So if you have TRMs generated throughout your life against different infections, now you're at a disadvantage that if one of them become reactivated because the peptide shows up somewhere else or there's same challenge elsewhere, now the ones in the brain can also react and induce this neuropathology, which is perhaps detrimental.
If you don't mind a follow-up question to that, so do you think, since you said the first early response is probably mainly to recruit peripheral cells, do you think in, let's say, disease where you have a virus that's periphery mostly and not entering the brain, would you then not see that response, do you think?
Yeah, that's a great question. So in our Tyler's experiments, we haven't seen that when we give the same virus peripherally. However, other people using VSV, so I think it would be virus specific in that case. I know John Hardy's lab in Iowa has used VSV peripherally, where they actually delivered it intravenously and they got TRM formation everywhere, including the brain and outside of the brain. There's also some interesting data coming out of COVID labs and hamsters that COVID infection can induce TRM activation and subsequent spike protein release due to another infection can reactivate them. So we think that's going to be virus dependent and it's actually pretty clinically relevant because if influenza, VSV, and COVID have all been shown to produce TRM activation in the brain, even though the viruses were not directly delivered to the brain.
Exciting, thank you.
Thank you.
So the next question is from Bebe Fan, and they say, great talk. First, how is the brain virtually memory CD8 T cell pool established in the first place? And second, are they HSC derived or fetal liver derived? And thirdly, where do the brain antigen-specific TRM get primed upon the new viral infection?
Very good question. So they are not virtual memory. We've done quite a bit of extensive characterization to actually show they're not virtual memory. So traditionally, virtual memory CD8s can have different markers. CD69 is possible, but they all express CD62L. Our T cells in the brain of a naive animal does not have CD62L. So we don't think they are virtual memory by that definition in the literature, but they are in the fact that they haven't seen a specific antigen before that we know of. Interestingly, in our studies and other people's studies, we don't know where they come from in terms of development, but we do know they're HSC derived, and you can actually establish them by bone marrow transplant into a rag knockout. So they are not fetal liver derived, and we've done experiment where we took a rag knockout, gave it bone marrow, and then it eventually did regenerate some TRMs in the brain as well. There are caveats with that model because irradiation can cause blood-brain barrier leakage and stuff, but I think it gets to the point that they're not fetal liver derived at least. So we don't know when they happen. We've looked at mice as young as 3 weeks and they have these TRM populations. We do know that they increase with age. So if we look at 1-year-old mouse, they have a lot more of them, even a naive 1-year-old mouse compared to a young mouse. But we still don't know exactly when they infiltrate the brain or what antigen they're specific for. Right now, we're doing studies to address that by using TCR transgenic mice like OVA as the previous speaker to actually see, even if in the absence of OVA, they can still respond. If that's true, they're truly non-antigen specific, they will go to the tissue. Maybe they respond to injury-induced cytokines, and then we will look into probably gamma chain cytokines. That would be my first usual suspect to get into for their responses.
And then Chunwei Chen asks, are there specific brain regions that brain TRMs reside in? Does TRMs translocate to different regions in inflamed brains compared to naive brains?
So that's a really good question. We haven't addressed that in our naive animals. We are planning some CyTOF strategies to actually look. It's very hard to do microscopy in our hands in a naive brain with that few amount of TRMs. But we're developing these mass cytometry tissue CyTOF basically to actually show that. So I don't know in the naive brain, but in the infected brain, and after infection is cleared, we do know that they are some hemispheric bias to where the viral infection was. So even in our MRIs that I've shown, you can see that the leakage is pretty localized to one side of the brain, which is the side we infected. So I would expect these TRMs would be mostly localized. However, you know, they have the potential to move. And I haven't verified whether they can go from one hemisphere to another anatomically. So that's a great question.
Okay, cool. So Xinjiang says, great talk, good job. And then Carolina Berta also says, incredibly complex and very engaging. Thank you for the talk.
Thank you.
We have, okay, we have one more question. So Samantha Golom says, great talk, thank you. Have you been able to do any IF or IHC on brains to look at the spatial distribution of the TRMs? I guess that's similar to the previous question about the brain region specificity.
That's a similar question. Yeah. We do think they are really in the parenchyma. We've done, I think, a good job with the IVs and then clearing to actually show they're in the parenchyma. So I don't think they're in the CSF or in the meninges, especially because we actually take the meninges out with the skull before we take the brain. So we are, sorry about this cat interrupting. So we have not really, we're sure they're in the parenchyma, but where in the parenchyma is, just like I said to the previous question, we're trying to analyze that with some multi-parameter IHC if possible.
Great. And then we have another comment from Chunwei Chen. Also, she says, great talk. And I think that's it. Are there any further questions from the audience?
Thank you so much for all the questions as well and the comments and posting. Thank you.